right, guys, we'll go ahead and get started. Good morning, everyone. How are you guys doing today? Good. All right, so just a quick reminder, I mentioned this in lab yesterday for those of you who weren't there. The final exam is going to be on May 2nd at, so it's a Thursday, it's going to be at 10:20. We have the room for 2 hours from 12:20. I assume it's this room, but I will double check on that and I will, let you guys know. We have 2 hours, but it's going to be a non-cumulative 4th unit exam, so on all of the lectures that we're covering, for this last unit. So let me know if you guys have any questions there. And so I guess today what we're going to talk about is we're going to start kind of going back into the immune system and talking about the adaptive immune response. So we talked about a lot of, microbial ecology and some immunology and the innate immune system, and we briefly touched on the adaptive immune system. So that would be that SWAT team analogy that I gave you guys. But today we're going to kind of put it all together and hopefully hone in on some of those concepts on microbial pathogenesis and the immune system and the innate and the adaptive immune system's roles. So just some basic principles of this damage framework hypothesis. So essentially our immune system is going to react, or it's going to be activated when there's some sort of damage by a microbe. And so microbial pathogenesis, being able to call it a pathogen, is an outcome of the interaction between a host and a microorganism. So we often call this a host-microbe interaction. And so the host relevant outcome of this host-microbe interaction is determined by the amount of damage that is done to the host. So you can't really call something a pathogen unless you know it's doing some sort of damage to the host, and in turn the host is responding through the immune system. The host damage can result from 2 things. The first being the actual microbe encoding factors like virulence factors, or you can have the host response being activated and causing some damage, and so that host response is to the presence of the microorganism itself. And so the host response can either be too weak or too strong. So if it's too weak, that can lead to disease. For instance, a weakened immune system that will allow opportunistic infections, those microbes that are normally not causing harm, but under some immune suppression or heavy antibiotic use, that can then allow these opportunistic pathogens to overgrow. You can also have too strong of an immune host response that can result in disease, and so we talked about this in the context of having a cytokine storm. So that's going to be detrimental to organs and can lead to death. You can have sepsis, which is a systemic response to high levels of toxins. So you can have bacteria that ends up getting into the bloodstream. We call that sepsis. You can be hypersensitive, and so that reaction, being really, really, really sensitive to a pathogen results in a really, strong immune response, and that can cause death. Any questions on this overview? Okay. So we've talked about this before. Pathogens have specific mechanisms that allow them to gain access to the body and colonize and adhere. So they basically are able to acquire the nutrients necessary. Then they have to multiply and persist, and then avoid the innate host defenses, and then also evade acquired specific immune defenses. So that's both the first innate host defenses is the innate immune response, and then also the acquired or the adaptive immune response. And then they're going to have to cause tissue damage and exit and transmit infection to the new host. So the first, 6 points here are causative agents of having like an infection. But when the pathogen is able to exit and transmit the infection to new hosts, or new cells, then that's when we can call it an infectious disease. So this kind of ties into what we talked about in infection versus infectious disease. So we'll go go through some slides on adaptive immunity. Okay, let's see if this will work. Let me make sure the volume is up. Just a quick little overview. Okay, I'll just do it this way. The volume up. Turn off the solar.
The Windows and Peelle.
What?
Windows Peelle.
What's Windows? Where?
Oh, the Windows.
Oh.
And then there's like speaker side on the right corner. And then there's like
This.
icon as
You want to come up here? Sorry. Make it easier. Oh. Oh yeah, that's what I do in the lab.
An overview of all the major body systems is nice, but something remarkable happens when you explore one body system at a time in a bit more depth. And, in our opinion, one of the most fascinating systems of all is the immune system. Why? Because it features cells in your body that work day and night together to protect you from a constant threat of pathogens trying to bring the system at a time in a bit more depth. Mayhem and an overview of all the major body systems is nice, but something remarkable happens when you explore one body system at a time in a bit more depth. And, in our opinion, one of the most fascinating systems of all is the immune system. Why? Because it features cells in your body that work day and night together to protect you from a constant threat of pathogens trying to bring mayhem. And most of the time, you don't even know it. Most of the time. By pathogen, I could mean a lot of things. A lineup, viruses, bacteria, fungi, protists, parasitic worms. We could go on. Your body has external protection against these things, like your skin. We call that a first line of defense because it's the first line against letting these pathogens inside your body. Mucous membranes too, like the lining of your nose, will keep pathogens from getting inside. This first line of defense is non-specific because it's not selective about what it blocks from getting into your body. But this is all not foolproof, and sometimes they do get in. And when they do, your immune system has all kinds of ways to deal. After all, this is not its first rodeo. So let's say they break through our first line of defense. Well, the second line of defense includes the inflammatory response. To explain it very simply, let's say you step on a sharp stick, and there's some bacteria on that stick, in your foot. The initial damage of this stick into your foot can cause certain types of cells, such as a mast cell, to react. These cells are filled with substances that work with allergic responses and inflammatory responses too. One substance that they contain is histamine. If they are triggered to release histamine, the result is that this will cause blood vessels to dilate, meaning they widen near the injury. Histamine also contributes to making these blood vessels leakier. The dilation and the leakiness of the blood vessel makes it easier for many types of white blood cells, such as certain types of macrophages, to reach the area. And macrophages do what macrophages do best. They consume the pathogens. Additionally, your body has a complement system. The complement system is not what it sounds like. It basically works to help or complement the actions of the immune system. It can work with nonspecific or specific responses. In this situation, the release of complement factors, in this case, can further attract macrophages to the area to consume pathogens. When all this signaling stops, the damaged area can return to normal. The pathogen has been terminated. However, that also was a nonspecific response. Who knows what was on that stick? So that takes us to the third line of defense, the specific line of defense.
That's adaptive.
If you had a cold virus spreading through your body, you may need your response to be targeted on that pathogen. Now, as we give our typical notice, the immune system is very complex. We've just been giving some basics, and we're going to continue to do so. But there are a lot of extra details and exceptions that this short video can't go into. We encourage you to explore. Okay, so we mentioned, what if we need a more targeted response? We're moving into something known as adaptive immunity. Sounds fancy. This is a specific response to an antigen. An antigen is something the body recognizes as non-self, and in this case, it is something that would be a part of the pathogen. This adaptive response is going to be the third line of defense, as the first and second line of defense may not have been enough to control the pathogen. We're going to focus on the basics of two adaptive responses, cell-mediated and humoral. Cell-mediated. This involves the cytotoxic T cell. The cytotoxic T cell is a white blood cell that has the ability to destroy cells that have been infected by the pathogen. It does this by releasing signals that causes the infected cell to do apoptosis, which is a type of self-destruct. It can do this by releasing a protein called perforin, which actually causes holes in the cell membrane. This causes water and ions to flow into it and destroys the cell. When cells that have been infected by a pathogen are destroyed, this can also destroy the pathogen, or it can mean the pathogen at least can no longer replicate inside that infected cell. The thing is, for this response to work, you have to activate a cytotoxic T cell. Stimulating a cytotoxic T cell could mean an infected cell presents an antigen from the pathogen that had infected it. The infected cell presents the antigen on its own cell membrane, kind of like a little flag saying, hey, I've been infected, and here you go, this is what it is. This activates cytotoxic T cells to bind and release signals that causes the infected cell to perform apoptosis. But there's another way to stimulate cytotoxic T cells too. Remember how macrophages may have been consuming the pathogen? When they do, they process the pathogen, and the antigens from the pathogen are transferred to the macrophage's surface. A white blood cell called the T helper cell can bind. The macrophage will release chemical signals, which then causes the T helper cell to release chemical signals, which then can stimulate cytotoxic T cells. Cytotoxic T cells will be in search of infected cells so they can stop the pathogen, and they will also continue to amplify the immune response. That's the cell-mediated response, simplified. But remember how we mentioned those helper T cells? They're big helpers, as they help not only in the cell-mediated response, but they also help in the humoral response. So what happens in the humoral response? In one scenario, a macrophage has consumed a pathogen, and once again, has an antigen from the pathogen on its cell membrane surface. Then it binds a helper T cell. That helper T cell could also stimulate a white blood cell known as a B cell. B cells are white blood cells that have the ability to make something called antibodies. Before I define antibody, can we just take a moment and recognize that there are three words that sound very similar and can involve the immune system. Antigen, which is something that the immune system recognizes as foreign to the body. We've been mentioning that one a lot. Antibiotics are substances that can specifically destroy bacteria. We have a separate video on those. But antibodies are something totally different. Antibodies are proteins, and they tend to be in a Y shape. Antibodies have an antigen binding area where they bind a specific antigen. They will be found in blood, but many antibodies can also be found in mucus, saliva, breast milk, and more. There are different classes of antibodies. For example, IgE can protect against parasitic worms, and it's responsible for a lot of allergic reactions. Antibodies are generally very specific, so there must be an antibody that is able to bind to an antigen. When antibodies bind an antigen, they can deactivate the pathogen by affecting the ability of the pathogen to move, reproduce, or cause harm. The binding can also be like signs telling macrophages, here it is, come eat it. So activating B cells causes these antibodies to be produced, and this is part of the humoral response. While B cells can be activated by a T helper cell, they can also be activated by free antigens themselves that they may come in contact with. Now, we do want to mention that both in the humoral and cell-mediated response, there are memory cells. There are memory B cells and memory T cells. These cells keep a memory of the antigen they were exposed to. Memory B cells can activate plasma B cells, which will make antibodies. Memory T cells can activate cytotoxic T cells, which will go after infected cells. The ability to keep a memory is very important, and this is also where vaccines come in. Vaccines can introduce either an inactivated or very weakened pathogen. This means the body does not get the disease itself, but it will launch an immune response. By launching an immune response, there will ultimately be memory immune cells that will be involved in launching an efficient attack if that pathogen is ever encountered in the future. Overall, this immune system that you have, it's pretty incredible. There are entire giant textbooks about this topic alone, and careers dedicated to studying it. Well, that's it for the Amoeba Sisters.
All right, so that was a really brief, but also, convoluted overview of a lot of the things that we're talking about today. So if you didn't, get all of that, that's totally fine. I'm gonna go through the slides and, discuss some of these things. So it would be really helpful for you guys to go back and rewatch the video after, you guys learn the material through the slides. And so it'll reiterate those concepts. The more you see it, the more you hear it, the easier it'll get for you to understand it. But I've shown you this before, a little overview of the immune system. I'm not going to keep, beating a dead horse here, but immunity is immediate. It consists of this non-specific cellular defense, and adaptive immunity is slower to respond, and it's reacting to specific structures, so it's a specific cellular defense, and it's reacting to antigens. So this is just another little image describing all of the team members within the adaptive immune response. You have helper T cells, the actual killer T cells, and B cells. But some key terms, we've talked about antigens a whole lot. They're foreign substances that cause the body to make an immune response against it. They can be toxins, chemicals, a bacteria, virus, or anything that comes from outside the body, so it's foreign. We're going to be talking about antigen processing, and so what that means is an antigen is broken down into its peptides, and it's being prepared for antigen presentation. An antigen presentation happens on antigen-presenting cells. So an antigen peptide that's processed is displayed on the surface of a cell that's an antigen-presenting cell. And the antigen-presenting cell is presenting it to the cells of the adaptive immune response, like B cells or T cells. So B cells are part of what we call humoral immunity. And so they are made in the liver before birth, and after birth, they're made in the bone marrow. And so after maturation, they migrate to lymph nodes into the spleen. And so these are antigen presenting, so they basically can also present antigens. They secrete cytokines, they express B cell receptors, and the main job of B cells to fight against infections is to make antibodies. And so there are two types. There are B cells that can differentiate into plasma cells. And so plasma cells are B cells that release antibodies in response to antigens. So once a B cell becomes a mature plasma cell, it can release up to 2,000 antibodies per second. And then we also have memory B cells. So these are B cells that remember particular antigens. So if they appear again in your body in the future, your immune system can then quickly mount a defense because these cells have some memory. So B cells after birth are made in the bone marrow while they're maturing, and so that's why they're called B cells. T cells are made in the bone marrow as well, but when they're immature and they're trying to mature, they leave the bone marrow and enter the thymus, and so that's why they're called T cells. And so T cells, like I've mentioned, can mature into different types. They can mature into cytotoxic T cells, and so their job is to kill, hence the word cytotoxic. So their job is to kill cells that are infected with the virus or bacteria or cancer cells. T helper cells, their job is to assist other naive T cells to mature and activate. And T regulatory cells, they regulate things. So their job is to keep the immune response in check. So we'll go through examples of these. But just as another, quick, image here, we have naive T cells. When they haven't encountered an antigen yet, they're still naive. We have regulatory T cells that modulate the immune system. There's also memory T cells, as well as the memory B cells that we talked about. And these also remember prior threats so that a prompt targeted response can be activated if that threat is returned. And then we have T helper cells and T killer cells. So this is the cytotoxic cells. T helper cells are sometimes called CD4 positive cells, and T killer cells are also called CD8 positive cells, and so we'll talk about that, but just keep those things in mind. So both B and T cells developed from progenitor stem cells. So the common lymphoid progenitor we talked about a couple lectures ago. So we have just regular hematopoietic stem cells in the bone marrow. And then they kind of split off into two lineages. One was the myeloid lineage and the other was the common lymphoid progenitor lineage. So the CLP lineage is what is giving rise to some immature, B cells, and thymocytes are what we call the first T cell precursors. And so those then can mature into B cells, T cells, and NK cells here as well. But they differentiate into these precursors after they leave after their common lymphoid progenitors. So that's why they are called pre-B cells and thymocytes, because they're precursors. So B cell development. B cell development can be broken down into steps with quality checkpoints. So B cells get their receptors in the bone marrow. We said that after birth, they are maturing in the bone marrow. And so their receptors include the rearrangement of genes that we call immunoglobulin, or IG. And so this will become a little bit more familiar in a second. But any B cells, there's a step-by-step process. So they get their receptors in the bone marrow. These receptors are specific for certain, antigens and things that they are meant to attack. But if a B cell has any receptors on it that are reactive to self, then those are identified and removed from that process. And so those are removed just by autonomous cell death, so apoptosis, and that's called negative selection. So your body, when it's developing B cells, it's going to go through a process to remove any that are going to react to self. And then the B cells that pass this checkpoint then can migrate to the lymph nodes, and so then they can bind antigens. And once they bind the antigen, they're considered activated. And then activated B cells can make plasma cells and memory cells. Any questions? Okay, so I'm telling you here that the B cells get their receptors in the bone marrow, and they basically are rearranging genes to end up having this immunoglobulin. So immunoglobulins are, in immunology, essentially, antibiotics are classified into several types of immunoglobulins, and so we call those isotypes, and so that's why here you see these 5 isotypes. But the antibodies basically have, are immunoglobulins, and they are Y-shaped. So this is what is produced by the B cell. It's always going to be a Y shape, and it's made of 2 polypeptide chains. So you don't have to really remember this. This is more in-depth immunology, but there is a heavy chain and a light chain. And so antibodies are produced by B cells or plasma cells, like we've said, and their job is to neutralize the pathogen. They also have roles in protecting the mucosal tissue and defense against parasites. And there are 5 isotypes of antibodies that trigger different functions. So that's where you'll see the immunoglobulins, IgG, A, M, E, and D. If you want to remember them, you can remember gamed. And So there's this variable region here, and this variable region near the tip of the antibody can differ from molecule to molecule in countless ways, allowing it to specifically target an antigen because they have to be specific. So different antibody isotypes trigger different functions. So when you see, antibodies, you should know that, we're talking about immunoglobulins. So there's a lot of terms that we're going to be talking about where we're using things kind of, I guess, like it's the same kind of name. Essentially, antibody isotypes and immunoglobulins are all referring to the same thing, but the isotypes here, they all have different functions. So you don't have to necessarily, actually you should probably know some of these, but I guess the important ones are remembering that IgE is responsible for allergic reactions, because that's something that's really important in allergies. And then, IgA we know is found in mucus, saliva, breast milk, tears. IgD, that's part of the B cell receptor. You don't really have to know that one. IgG is secreted by plasma cells in the blood, and it's important really in when in fetal development because it's able to cross the placenta from the mom, and that's how the mom gives the fetus antibodies before they're even born. It's through this IgG. And then IgM can be attached to the surface of the B cell or secreted in the blood, and that's also responsible for early stages of immunity. And you notice they all have Y shapes, but they all are different. They have different structures. This one's a dimer. This one has 5 of them attached to one. But the important thing here is that these isotypes can switch back and forth and make different, more different isotypes, but this is influenced by cytokines. So we talked about cytokines last lecture being these messages for the cells. There are these chemical signals that tell immune cells you need to come and attack, or there's inflammation here, and there's different cytokines that are associated with different immune responses, and so it's just really important to recognize that different cytokines will either inhibit or induce different isotypes. And so we call this isotype switching. And so B cells, we talked about them either differentiating into plasma or memory B cells. And so that process also happens under the influence of different cytokines. So cytokines really control a lot of who's coming to the site, what they're producing, what type of response. So they are those chemical messages, and they're coming up here again as being really important in actual B cell development. Any questions so far? So this is an antibody, and here we have this hypervariable region. So that just means that this is the region that's going to vary between different antibodies, and that is going to be what binds to different specific antigens. So this area we are calling the hypervariable region. Essentially, this would be the B cell receptor. So this is essentially the antibody receptor, and its function is to bind to antigens. They can bind to whole antigens or just fragments. So here I'm showing you a whole antigen, but this antigen has specific epitopes or structures that will bind to certain antibodies. So if you have an antigen with more than one epitope, then that would be multivalent. So this one has 2 epitopes that are binding to these antibodies. So this means that it's multivalent. And so each one of these is an epitope. Does that make sense? These are 2 different antibodies. And the goal, again, of antibodies or B cells is to neutralize the pathogen, and so that way they prevent cell entry, and or they can facilitate phagocytosis by phagocytic cells. So that's a quick overview on B cells, their receptors, their function. They neutralize pathogens. Any questions on this before I move on to some T cell overview? Okay, so T cells, in the video that was called cell-mediated immunity. Reminder, B cells, that's called humoral immunity. So in this cell-mediated immunity, T cells are made in the bone marrow as well, but they end up leaving the bone marrow and going into the thymus to mature. And they can mature into different types. Once again, that's the cytotoxic T cells, or CD8 positive, whose job is to kill, T helper cells, CD4 positive, and then T regulatory cells. And so, once again, I have this up. And then, once again, the different types of T cells here. But what is the thymus anywhere? So the thymus is over here, right below your trachea, in between your lungs. And so this area is essentially a boot camp for T cells. So T cells, they're in the bone marrow, and they move to the thymus when they are being, when they're maturing. And so basically in the thymus, they're being tested to make sure that they can correctly bind to MHC. We'll talk about what that is. And so basically, they have to go through this check as well, as quality control check, to make sure that they won't attack your body's healthy cells. So just like B cells, they also have to undergo these checkpoints to make sure there's no reaction to self. And so T cells, where do they get their receptors? CD4 T cells get their receptors from helper T cells, and CD8 T cells are the cytotoxic T cells. So the receptors on them are the CD4 and the CD8. So those are right here. So the CD4, that's the receptor, and T cells, the receptor here is the CD8. And so only the T cells that pass the test are released into the body. And then one thing that is important is the thymus is actually larger in children, and it gets smaller as we age. So that important, immune cell development in the thymus, the T cell development, is really important when you're younger, because when you age, you end up graduating fewer T cells, so basically mature T cells. So at the age of 20, that's when you start to actually have less and less T cells maturing in that area. So that's why having memory cells is really important. And so lymph nodes are tiny organs that filter lymph fluids in our bodies. Lymph fluid is the collection of fluid that drains from your cells and tissues, and there are hundreds of lymph nodes throughout the body. This picture just kind of gives me goosebumps, because literally everything is connected in your body through the lymph nodes. And so basically the lymph node functions as this watchtower, or this elevated observation point, and it's really like the headquarters for infection and recognizing it. And so it provides a way for the immune cells to monitor the entire body. And reminder, the body has lymphoid tissues. So lymphoid tissues are, tissues that have connections to lymph and blood vessels. And so the primary lymphoid tissues are that bone marrow, the thymus, and secondary lymphoid tissues are the lymph nodes, your tonsils, your spleen, Peyer's patches, and those mucosa-associated lymphoid tissue, or gut-associated lymphoid tissue, or MALT or GALT. So I guess in this context, we're talking about all of these lymphoid tissues being a place where we have these immune cells. And so I guess in the context of people who have to remove their tonsils, do you think that would decrease your ability to fight off infections, or will it increase your ability to fight off infections if you remove them? Like, how do you navigate that, I guess? Kind of a double-edged sword. There's no one, correct answer. So basically, there's a lot of people, and even children, who will get recurrent tonsillitis, and they have to remove their tonsils. So basically, it comes to the point of the physician or the surgeon asking, is it worth it to, if you're removing your lymph nodes, you're removing part of that immune system function, fighting against things. But if your tonsils, for instance, are getting recurrently infected, and it's not going away, and you try antibiotics and they end up getting resistance, and things like that, then at that point it's worth to just remove the tonsils, because you have a lot of other lymphoid tissue in your body that can protect against things. You also have adenoids and things like that. So it's really a double-edged sword, because you're losing something, but sometimes it can drastically change somebody's life to remove the thing that's causing the problem. Sometimes, you know, people that have their tonsils end up, you know, living better lives without them, because they no longer have the thing that's harboring the infection anymore. So T helper cell types. So we talked about the helper T cells, and there are different kinds. So T helper cells can be within different classes, so Th1, Th2, Th17. You've probably heard a little bit about this before, but the Th just means that they're all T helper cells. Th1 cells help macrophages. You don't have to remember which cytokines here are associated with Th1 cells, but essentially in the immune system, if we're trying to measure, you know, what type of response is happening, we can measure the levels of cytokines that are being produced and then attribute those cytokines to a specific type of response. Like, is it a Th1 response? Is it a Th2 response? Th17, that kind of thing. So we read these messages from cytokines in order to understand what the immune system is doing. Is it T helper cells that are trying to help macrophages? Is it, you know, Th2 cells that are causing these mast cells to come up, and things like that? So Th1 cell helps macrophages, and Th1 responses are usually associated with inflammation. Th2 cells are usually associated with allergic reactions, and so when you see eosinophils, basophils, and mast cells, you know, mast cells especially are very much involved in allergic reactions, and so Th17 help neutrophils, and then there are these TFH cells that help activate naive B cells, and then Tregs, so they're regulatory T cells. These control and limit the activation of other CD4 and CD8 cells. So basically, these just help regulate things and control things so you don't have too much of a good thing. And once again, cytokine messages influence which subtype of T cells the T cells are differentiating into. So that's how, you know, some of these are fate specifying. If you have an IL-4 cytokine being produced, then, you know, and these other cytokines, these are all associated with Th2 cells. So in my research right now, I'm looking at the effect of amoxicillin on the immune system. So what I did was I basically, you know, did some assays and methods to eventually measure the amount of cytokines that are expressed in the host tissue, and then I'm able to make conclusions about what type of immune response occurs when you are taking antibiotics. And so is it inflammatory, is it not, and things like that. And so I want to, you know, back up a little bit. You know, we talked about T and B cells, but these are, you know, T and B cells are part of the adaptive immune response. And so there has to be something that's, you know, presenting the antigen to the T and B cells in order for them to come and do their thing and activate their response. So we need these antigen-presenting cells that are part of the innate immune system. So dendritic cells are key antigen-presenting cells that we talked about, and so they, you know, they inform other, you know, cells like T and B cells to fight against pathogens that invade and surpass the dendritic cell. And so these are a distinct type of white blood cell. They also can process antigens, so that's the antigen processing, where they break them down into peptides. And dendritic cells can exist in two distinct forms, like other cells as well, immature and mature. So here, an example of how adaptive immunity works. So you have here an epithelium, and in red we have a pathogen, and the dendritic cell here in purple. And so the dendritic cell binds to the pathogen, or this is a virus, and so it's basically infecting and replicating within the epithelium, and so the dendritic cell picks it up. Once it picks it up, the dendritic cell becomes activated. Notice here that there's inflamed tissue because the dendritic cell is probably producing cytokines and things that are, trying to signal there's something going on here, so we need an inflammatory response. And so the dendritic cell then takes the infection to the lymph node, where in the lymph node you have these T and B cells that are immature, and they get primed and they mature when this dendritic cell basically presents the virus to these cells. So it can present it to B cells or T cells. They get activated. Here the T cells just getting activated, or the B cells becoming plasma cells. So here in the case of the plasma cell, that can then create antibodies because it's a B cell against the virus, or if the T cell is activated, the T cell here can just attack the virus and kill it itself. So kind of going from innate to adaptive, and the role of B cells and T cells, kind of a quick overview of how that occurs. Does that make sense? Any questions so far? Okay. So I mentioned MHC, the major histocompatibility complex. And so this is the most important genetic component of the mammalian immune system. And what it is it's basically a large region on the DNA that contains a set of closely linked genes that have just many different forms. And those genes code for cell surface proteins that are essential for the adaptive immune system. So basically, the MHC are these genes within our genome that encode for proteins on the cell surface of the T cells. So these cell surface proteins we call MHC molecules. So MHC, remember we talked about the CD4 receptor on T helper cells and the CD8 receptor on cytotoxic T cells. MHC binds antigen peptides from the pathogen and displays them on the cell surface so that the appropriate T cell, whether it's the CD4 receptor or the CD8 receptor, can then bind to that antigen. So MHC is what's kind of binding the antigen. So I just told you that antigen-presenting cells can present antigens to T cells, and now I'm telling you that MHC molecules bind antigens and they give them to T cells. So kind of let's link that all together. And so essentially, me saying that there are a set of closely linked genes that have many forms, that means that they are polymorphic, which means you have multiple variants of each gene within the human population. And we want all these different variants because we want to recognize different antigens. So MHC molecules are required for T cells to recognize foreign antigens. There are two types of MHC molecules. Type 1 is the one that is presenting antigens to killer T cells or CD8 positive cells. This one targets mostly intracellular antigens, and I'll show you more of this shortly. Type 2, these are expressed on antigen-presenting cells. So basically, the MHC molecules are expressed on the antigen-presenting cells, like dendritic cells and things like that. Type 1 antigens are expressed on most cells. Type 2 molecules are expressed on antigen-presenting cells as well. But type 2 presents to the helper T cells or CD4 positive. And these ones target mostly extracellular antigens. So here, what I'm showing you is this MHC class 1. So this is the molecule that's on the immune cell, and this is the same thing, MHC class 2, and they bind the antigen peptide, and they basically show this to either a CD8 T cell in this case or a CD4 T cell in the MHC class 2. But these molecules are required for the T cell to be able to recognize antigens. So T cells can only recognize pieces of an intruder when they are bound to MHC molecules. It's just me saying the same thing in a different way. The binding of the T cell receptor to the MHC molecule that's containing the antigen peptide is usually a little unstable, and so some co-receptors are required. And so the CD4 co-receptor is expressed by helper T cells, and the CD8 co-receptor is expressed by cytotoxic T cells. So we have these MHC molecules, and they have to bind to that receptor, either the CD4 or the CD8, that is on the actual T cell, whether it's helper or cytotoxic. Any questions, confusions at this point? Okay. So activated T cells have to travel to the site of infection once they're activated, and so here I'm just showing you again that different types of cells, T cells will elicit different cytokine production. And so CD8 T cells, they're cytotoxic. They can kill any infected cells that they contact. CD4 T cells act a little bit more specifically in that they can interact with a wide variety of cells. But yeah, so just kind of keeping in mind that T cells can only recognize a piece of the foreign antigen when they're bound to MHC molecules, and those MHC molecules are binding to the CD4 or the CD8 co-receptor on the T cell. So I'm going to kind of put this all together and give you an example of dendritic cell antigen presentation and T cell activation. So there are two major steps to T cell mediated immunity. Naive T cells have to be activated by dendritic cells in the secondary lymphoid tissues. And then the T cells that are differentiated can then travel to the infected tissue, or they stay behind and they act locally, but they're naive T cells until the antigen presenting cell activates them. And so here, these are just different ways that pathogens are infecting, and just roles of how the antigen is being processed by a dendritic cell. So there's just different ways that a dendritic cell can process different antigens depending on whether they're bacteria or viruses, and then which MHC class molecule is loaded. But just notice that anytime the MHC molecule is class 2, it's going to basically activate CD4 T cells. If it's class 1, it's going to activate CD8 T cells. So trying to combine all these concepts together, looking at the system at work, I want you to focus your attention over here. So you have your antigen presenting cell, which is the dendritic cell. So here we have our antigen presenting cell. This is the microbe in red. The antigen presenting cell, I guess since it's engulfing, we're going to call it a macrophage, not a dendritic cell. And so it's going to engulf the microbe at the site of infection. It's going to digest the microbe and the antigens, and it's going to present the antigen on the surface. This antigen can then be, presented to B cell, because some of them, they're just broken down and released, and so they are free floating, and so they bind to the B cell receptor. And then B cells can be differentiated into plasma cells and secrete antibodies. So this is T cell independent, because that has nothing to do with T cells. The B cell is being triggered by the antigen that's free floating. It's not a helper T cell that's giving it to the B cell. So this one is T cell independent. So that will trigger differentiation of the B cell to plasma cells and the secretion of antibodies. On the other hand, you can have this antigen that's presented on the antigen presenting cell. This antigen is going to be, if it's being basically taken to a helper T cell, would this have CD4 or CD8 receptors? So if the antigen is being presented to a helper T cell, do helper T cells have CD4 or CD8? It would be MHC class 2, so it would be CD4. So CD4, and then this then is another T cell that it's, helping out, I guess. It's another helper T cell, but this is a T cell dependent antigen because it's depending on the T cell to take it to the B cell so that the B cell can then differentiate into plasma cells and secrete antibodies. You can also have, independent of any B cells, you can have the antigen presenting cell, so this macrophage, giving the antigen or presenting it to a cytotoxic T cell. So now we're entering just cell-mediated immunity. And then this cytotoxic T cell is activated once it binds to the antigen, and then it can ingest that antigen, takes it up, and then it can kill, because that's what cytotoxic T cells do. They basically circulate and find and destroy the infected host cell. Any questions? So once again, lymph nodes function as the rapid response center or the headquarters during infection. So another way to show you this, here we have the antigen presenting cell that's engulfing the pathogen at the site of infection. And so then these antigens and antigen presenting cells can then enter the lymph nodes or lymphatic vesicles and then present the antigens on their surface and move into lymph nodes and where T cells and B cells are. In the lymph node, these free antigens can interact with B cells, so this would be T cell independent in this particular case because they're free antigens. Or you can have the APCs engaging with T cells and activating either B cells for plasma cell differentiation and memory cell differentiation, or the T cell can then also just mature into the cytotoxic T cells. And so those enter circulation, and so they're really all over in your body. And then plasma cells and memory cells enter the bone marrow, and because that's where they go, and then the cytotoxic T cells can migrate to the infection site. So lymph nodes really connect everything, and they function as this rapid response center during infection. So clonal expansion. We said, cytokines are produced by these T helper cells. Here it's called T4, so that's another way to remember that it's CD4 positive cells, and they bind to MHC class II molecules. And so these cells can help activate B cells to rapidly proliferate. So this is me giving you this example here where it's T cell dependent, where you have a T helper cell that takes it to the B cell. So cytokines are produced during this process. And so basically when these T helper cells help to activate B cells, the B cells that are activated can rapidly proliferate and produce a large amount of clones of thousands of identical B cells. And so in this way, even though only a few B cells in the body may have an antibody molecule that's able to fit that particular antigen, eventually you have just so many clones and expansions that make thousands of cells produced with that right specificity to the antigen. So that's how we make memory B cells. It's called clonal expansion. All right, so here's a scenario. There is a Streptococcus pyogenes infection in the pharynx, so in your upper respiratory tract. And here we have a child. This is their pharynx epithelium, and this child has antibodies against S. pyogenes, so Streptococcus. So here we see the Streptococcus in red and the antibodies that are in yellow. And if a child already has antibodies that are made by these B cells in the pharynx area, then those antibodies are going to be specific and immediately just tag all of these all of these bacteria. So they're preventing the attachment of bacteria to the tissue, and most of them are then swept to the gut. And so that's where bacterial populations is limited and kept at a steady state, and the child remains healthy. But this child has a brother that doesn't have these antibodies, and so what happens then is the bacteria can stay in the pharynx and multiply and attach to the epithelial surfaces, and that bacterial population will expand out of control and cause damage to its environment, so the brother will suffer with a sore throat. In the context of a viral infection, here we have an adult with anti-influenza virus IgA antibodies. And so the virus is just attacked with all these IgAs, and so it can't infect cells. Because remember, a virus needs a host to replicate a biological host, and so if it can't bind to the host, it's not going to be able to infect cells. And so this adult remains healthy. Twin sister doesn't have the anti-influenza virus antibodies, and then the virus infects the cells, and this twin sister gets severely sick for 2 weeks. So that's why you get vaccinated, is to make sure that you have certain antibodies to protect against infection. Questions so far? Okay. So pathogen infection sites. So the places that a pathogen can infect are inside of the cell, outside of the cell, and if it's inside of the cell, it can be in the cytosol, or it can be actually like inside of the cell, inside of a vesicle. So just to basically remind you here, I'm showing you, this is cytosolic pathogens. Any cell can attack a pathogen, here shown in red. A macrophage here is what is going to engulf a vesicular pathogen, something that's in a vesicle. And so just a reminder here, macrophages are part of the innate immune system. And then here a B cell is what's going to create antibodies and attack extracellular pathogens. So just to remind you, the cells of the innate immune system, we've talked, about a lot of these, short of a few, like NK cells, but macrophages, eosinophils, basophils, mast cells, neutrophils, dendritic cells, these are all cells of the innate immune system. And the antigen-presenting cells that present to the adaptive immune cells are these two that we talked about, macrophages and dendritic cells. So just as a reminder, so cytosolic pathogens, they're degraded in the cytosol. These ones can be killed by any cell. So the peptides are binding to an MHC class 1 molecule on the cells and presented to effector CD8 T cells, because MHC class 1 binds to T cells with the receptor of CD8 positive. And so because they're cytotoxic and it's undergoing a class 1 presentation and CD8 T cells, you're going to get cell death. So these are cytotoxic. If you have a pathogen that's inside of a vesicle, you're going to have antigen presentation by a macrophage of the innate immune system. These will have MHC class 2 on them, so they will present to these helper T cells or these CD4 T cells, and they then activate to kill bacteria, parasites, and they have other methods of then helping and taking the antigen to other T cells. And if it's extracellular, then that's going to require presentation either just directly B cells that are eliminating it by producing antibodies, or you can have antigen presentation through MHC class 2, so it presents it to CD4 T helper cells, and the T helper cells can then take it to a B cell to eliminate those extracellular pathogens through antibody production. All right, let's see. I have a few more slides. How are you guys feeling? Do you guys feel like it would be helpful for me to stop here and go through some of this again next time? I just want to make sure you guys are really getting this, and then we can finish off next time and reiterate some things, or, yeah? Okay, that sounds good. Then we can stop here, and then on Monday, try to review this on your own, because next time it'll, be a lot easier to grasp everything. And so, yeah, we can just continue on next time.